Welcome to today's lecture on embryology and neurodevelopment. In this session, we will be delving into the intricate world of neuronal development and exploring the various signaling pathways, molecular mechanisms, and cellular processes that shape the growing nervous system. From the initial patterning of the neural tube to the complex formation of synaptic connections, we will unravel the mysteries behind the remarkable development of the brain and spinal cord. So, let's get started. The formation of the neural tube, a crucial step in central nervous system development, involves sonic hedgehog signaling. This pathway influences the patterning of the neural tube through the interaction between sonic hedgehog protein and its receptors. Activation of this pathway leads to the differentiation of distinct cell populations along the dorsoventral axis, contributing to the development of different neuronal populations and axonal pathways. Defects in sonic hedgehog signaling can result in serious developmental abnormalities. The WNT signaling pathway plays a crucial role in the differentiation of neural progenitor cells into neurons. It activates the expression of proneural genes and influences neuronal morphology. Dysregulation of WNT signaling is linked to neurodevelopmental disorders. Ongoing research aims to uncover the underlying mechanisms. Proper migration of neurons during development is guided by F receptors and ephrin ligands. Their interactions can repel or attract migrating neurons, ensuring they reach their appropriate destinations. Disruptions in this pathway can lead to neurodevelopmental disorders. During neural development, axon guidance involves intricate molecular mechanisms that enable growing axons to reach their correct target cells by detecting and responding to extracellular guidance cues. These cues bind to receptors on growth cones, triggering signaling pathways that regulate axon motility and navigation. In the video, we will discuss the process of synapse refinement in the developing brain. This involves the elimination of excess connections and strengthening of active synapses through activity-dependent processes. Various molecules and cellular processes, such as trophic factors and astrocytes, play important roles in this refinement. These mechanisms are critical for the proper functioning of the nervous system and may provide targets for therapeutic interventions in neurodevelopmental disorders. Myelination, a key aspect of nervous system development, is regulated by genes and can impact disorders like multiple sclerosis and leukodystrophies. Understanding this genetic control can inform treatments for myelin-related conditions. Neural circuits undergo precise sculpting and pruning during development. Programmed cell death, or apoptosis, helps eliminate excess or poorly connected neurons. Various mechanisms regulate neural cell death, including neurotrophic factors and neuronal activity. Imbalances in cell death can lead to neurodevelopmental disorders. Neural circuits have the remarkable ability to adapt and rewire in response to environmental stimuli or damage, known as neuroplasticity. This process is involved in learning, memory, and recovery from injury. Synaptic plasticity, specifically long-term potentiation, LTP, and long-term depression, limited, is a fundamental mechanism for this adaptation. Neuroplasticity involves molecular mechanisms such as changes in gene expression, synaptic protein turnover, and alterations in neurotransmitter release and receptor function. Neurotrophic factors also play a crucial role in promoting plasticity and functional recovery. Understanding these mechanisms offers opportunities for interventions to enhance brain repair and recovery in conditions like stroke or traumatic brain injury. Exploiting nervous system plasticity is a promising approach for regenerative medicine and neurorehabilitation. During the critical periods of visual system development, synaptic plasticity is highly important for optimal learning and the formation of visual circuits. Molecular mechanisms involving factors like GABA, CSPGs, myelin-associated proteins, and neurotrophins regulate this plasticity. Disruptions in critical period timing or plasticity can lead to visual impairments, but studying the molecular basis of critical periods could help develop interventions for visual deficits. Rett syndrome is a severe neurodevelopmental disorder predominantly affecting females. It is characterized by a loss of purposeful hand skills, impaired language development, intellectual disabilities, and repetitive hand movements. The genetic basis of Rett syndrome involves mutations in the methyl CpG binding protein 2, MACP2, gene located on the X chromosome. MACP2 is essential for normal neuronal development and function. MACP2 is known to regulate gene expression by recruiting co-repressors and modifying chromatin structure. Dysregulation of MACP2 leads to altered expression of numerous genes involved in neurodevelopment and synaptic function. Although there is currently no cure for Rett syndrome, ongoing research aims to understand the molecular mechanisms underlying the disorder to identify potential therapeutic targets. Strategies such as gene therapy and pharmacological approaches hold promise for future treatments. This video explores the collaboration between neurons and glial cells, focusing on astrocytes and oligodendrocytes. It discusses their roles in synapse development, maturation, and function, as well as the consequences of disruptions in these interactions. Summarized version: stem cells show promise in repairing and regenerating damaged nervous tissue. Different types of stem cells can differentiate into neurons, astrocytes, and oligodendrocytes. Challenges include immune rejection and ensuring successful integration and function of transplanted cells. Research continues to explore their potential for nervous system repair. In conclusion, the development of the nervous system is a complex process involving signaling pathways, molecular mechanisms, and cellular interactions. We have explored topics such as signaling, axon guidance, and neurodevelopmental disorders, gaining valuable insights. Further research will enhance our understanding and drive medical advancements.